Dr. Tal, welcome to the Tick Bootcamp podcast.
Thank you very much. I'm glad to
Well,
be here.
actually, I guess it's actually welcome back. We had,
It
or at
is
least
welcome back, Rich.
Matt had the pleasure of interviewing you right after or right before we walked the runway together. So I guess we should first introduce ourselves as fashion models and then as Lyme geeks. And I was sharing with you offline that today is Matt's geek birthday because he has a birthday present and having you on our podcast today. So thank you for rejoining us.
Well, thank you so much for having me. I'm delighted to be here.
So I'm looking forward to seeing you in action, not just as a model, but now as a scientist. So why don't we build that background up for our folks? So where are you calling in from, and give us some of your background.
All right. Well, I'm calling in from home at the moment. I live in the woods, just not too far outside of MIT. Um, I my background.
Well, I mean, so again, we can't gloss over MIT. So you are a professor at MIT. So talk to us about that. So what are you a professor of, and how long have you been a professor at MIT?
So actually, I am a principal scientist at MIT. It means I have my own lab. I get to direct research. I'm also the associate scientific director of the Center for Gynepathology Research at MIT. I've had my own lab at MIT for over 4 years now.
Well, congratulations.
Inching towards 4.5. Thank you. Yeah, that feels like a milestone. So I'll
So
take
is it
that.
called the Tall Lab?
The Tall Research Group. Yeah.
Oh, cool. That's really cool.
Yeah, TRG. Um, so we yeah, basically I had been working at Stanford as a postdoc and then as an instructor, and I started to have some really big questions about Lyme disease that I really wanted to pursue further. I felt like it was only about year 4 of my postdoc that I started asking the right questions, and that's when I stumbled on chronic Lyme. And I really felt like these were some really important questions that, in immune regulation and immune responses to infectious disease, that just I felt like there was a lot of low-hanging fruit, just there for the picking, really, as happens in stigmatized diseases where people stop asking important questions and start making some assumptions that maybe aren't correct. And so
But wait,
that's
let's
what
pause
started
there for a
that.
second because I want to leave that as a teaser. We're
Okay.
already very excited about these questions, but give
All
us a
right.
little more background on the work that you were doing at Stanford and what you were doing your postdoc in. And so build out your education and what you were
Okay,
doing
great.
before you arrived at MIT.
Sounds great. I'm going to go one step back then. So I did
Okay.
my PhD with Akiko Iwasaki at Yale, and she's the one who really got me started to think about these interactions between the host and the pathogen, and how the host recognizes that there's an infection, how the host is responding to that infection. And so my early work in Akiko's lab was focused on viruses and how viruses are detected, and really around how the mitochondria are involved in that signaling process, and what can go wrong when your normal repair and maintenance processes are dysfunctioning, and then mitochondria are dysfunctioning, and then how that would impact the host overall, and specifically immune signaling. And the funny, ironic thing is that while I was doing that research, I felt like I was doing very basic science, and that I wanted to do translational immunology that would really impact human disease. And so I pursued a postdoc with Irving Weissman at Stanford, so that I could really get into applied immunology. And that's really where my career started as an immunoengineer. But while I was doing my postdoc on immune regulation and how these checkpoints are put in place so that your immune system doesn't kill you in the attempt to kill an invading pathogen. While I was doing that COVID happened. And suddenly, my PhD research on a virus that was impacting mitochondria and having all these, catastrophic effects was suddenly not so basic and suddenly quite relevant. And so I was suddenly found myself in a space where, um, you know, journalists were contacting me to weigh in on different things. Early in COVID, I found this, voice on Twitter all of a sudden, and so, there was an interesting irony there. One of the reasons I was so interested,
Or maybe not an irony. Maybe it's
or maybe
what you
basic
were
science just
created to do.
yes, yeah, no, I actually really do feel a huge sense of purpose in the work that I do, because when I started working on chronic Lyme, so in my postdoc, I was working on this checkpoint. So an immune checkpoint is this kind of it's a brake on the immune system, and it's meant to throttle the immune system intentionally, because your immune system can kill you, but obviously evolution would not have favored for our immune systems just killing us. And so
Okay.
there's these different throttle points, these checkpoints. And I was working on a particular checkpoint that was, there were all sorts of indications that pathogens have been manipulating this particular checkpoint. And I started looking at pathogens, and I walked the halls of Stanford microbiology, and I was asking everybody to test a probe that I had. And one of the pathogens that just lit up for us was Borrelia burgdorferi, the bacteria that causes Lyme disease. And that was the first I really thought about it. Even though I had spent 6 years at Yale in Connecticut, laying out in the sun on the grass, I hadn't given Lyme disease much thought. I'd heard about it, but now it popped up on a screen. And now I saw that it was manipulating the checkpoint, the immune checkpoint that I was working on. So
Okay.
I
So
start
I'm
reading.
going to skip pause again
Okay.
because this is all really exciting, but I want to go a little bit more into your background, and then we're going to need a little bit more background on the immune system, because we have a very broad spectrum
Mhm.
of folks who listen to this podcast. We have people who think they may have Lyme disease, who are listening to see if they can gain some insight into how they're feeling and whether or not they have Lyme disease. We have people who have just recently been diagnosed with Lyme disease and know nothing about the immune system. We have people who are supporting people who have Lyme disease, who are trying to
Yeah.
learn about the immune system. And what we're doing is we're at a very, very high level right now. I'm going to walk it back, but I want to walk you back a little bit more. So where did you grow up, and where did you go to college? Where did you do your undergraduate work first?
Okay. All right. So my undergraduate was in Israel. So let me go way back. So I'm born
That would
in
be great.
Israel, um, on a kibbutz,
Really?
in northern Israel. Yep.
Oh,
Hmm.
cool.
Yep. My parents, decided to move to a kibbutz to raise their family. That's where I was born. We moved to the United States when I was 6 years old, and I got to do first grade, first grade through 12th grade in Connecticut, actually.
Really?
And
No coincidences.
yeah, I then went back to Israel. I did my military service, which is, it's a civilian military, so that was required of me. And I afterwards stayed for university. I actually met my husband in the military, and so we did our undergraduates together at Ben Gurion University of the Negev. It was a really great place to study biology, and I really got very, very interested in immunology and microbiology at that time. And so I knew that was something I really wanted to do. I had often wondered if I wanted to go into medicine or medical research. It was always kind of tipping between those. I had some very formative experiences. When I was 8, my uncle died of AIDS, and at a time that we knew very little about HIV and what was happening, and And then when I was a senior in high school, my cousin died of meningococcal meningitis. And I saw my aunt and uncle go through the whole process of learning that there was a vaccine for that. And, just the whole, that whole process of just when you realize that, that's kind of the cost of these, there's vaccine preventable illnesses. And so that hit really deep in kind of my thinking about these processes and how these, responses to infection can be engineered and can be modified and can be prevented or treated, and we can change these. So I think I never really stopped thinking about that. And yeah, so when I went into college, I was very interested in both microbiology and immunology, looking at both sides of this. And then it was really immunology that caught my I was just so fascinated with the immune system. And so I applied to Yale and managed to get in, which was really great. Actually, both my husband and I got into Yale for graduate school. And
Congratulations, that's cool.
thank you. That was amazing. That was amazing. And I
It's
studied
good to learn that you didn't
immunology.
marry down. He got into Yale as well. So I teach us a little bit more about why you were so interested in immunology. You had these personal life experiences,
Yeah.
and there were some signals to you about both microbiology and immunology. You studied both as an undergrad, and the field that really caught your eye, the thing that your passion was calling you to study was immunology. First of all, what is immunology, and why do you think that was the field of study that caught your eye?
Yeah. So immunology is the entire system that you have to fend off invaders, and to repair and essentially to sense different damage of different sources and to try to, to attempt to repair that damage and get you back to your normal baseline state. And so this process, this system of the immune system, this just really captivated me as an engineering opportunity. I think before I realized I was an engineer, I never became an official engineer, but at MIT, I'm in the School of Engineering, and they asked me when I was applying why I wasn't applying to the School of Biology and why the School of Engineering. And I was like, I like applied immunology, which makes me an immunoengineer. But I really am so drawn to systems that we can engineer and to think about things like an engineer and how can we fix this and how can we make it better.
Okay,
So
so
yeah.
and how are you going to? So you now define the immune system for us. Define engineering first. How are you defining engineering?
Okay. I'm defining engineering as a way of looking at a system, thinking about how to make it better, thinking about how you can fix it, thinking about how it works, essentially that take it apart and put it together and make it work even smoother. That's how I think about engineering, and that's very much how I approach my study of immunology.
Okay, that's really cool. So let me, and I don't want to go off on too many tangents because I still want to build out your background because I'm already fascinated. Would it be fair to define humans as bags of bugs? And would it be fair to define the immune system as that system that manages the bag of bugs so that we are not being injured by the bugs that are either coming into our system or are already in our system?
So I think bag of bugs would be the funniest description I've ever heard for your colon. I'm not sure I would say it's fair to describe the entire body that way, but I would absolutely give you that your large intestine could be defined as a bag of bugs. And I
Okay.
think that's really, really funny. I love it, actually. I love it. Um, I've once heard another engineer describing that part of the body as, three balloons right next to each other, your bladder, your colon, and your uterus, for those of us who have them. And I thought that was so, I love these kinds of ways of thinking about the body. So a bag of bugs, I do think about it as a tube. So you have this inner tube, which is entirely open to the outside world, right? And non-sterile things come in, and there's all these bugs that live with us along this tube that
And how
goes.
do how do the bugs come into us? What are the different avenues of bugs coming into the system?
Love it. Okay, so this tube goes from your mouth and nose out through your anus, right? And, primarily the ways that we are going to introduce most of these bugs is either through breathing them in through our air, or getting exposed to them orally. And so I often think one of the most important aspects of your education is actually long before you start school. It's when you're a toddler, an early toddler, and just sticking everything in your mouth to learn your environment, to become tolerant to your environment. So you are actually training your immune system to not respond to food and to things that you're exposed to through your mouth. Because obviously that would be very, very bad if you start reacting to everything that you eat, which can happen in some disease states when that tolerance breaks down. So, but yeah, this is a major part of your education. And
So
so
so there are bugs all around us,
there's
right?
bugs all around us.
So every time we
And
breathe,
we
we're
are not
bringing bugs
sterile. There are
in,
bugs in us.
right? All right. So every time we breathe, bugs are coming in.
Yep.
Every time we eat something, bugs are coming in.
Yep.
Every time we touch our nose or our mouth or our eyes are going to be a place where bugs can come in. Can bugs come
That's
into
it.
our ears too?
Normally, I mean, they could. That's not a normal route because of you have this membrane, your eardrum, and that would not be a normal route unless that was um,
Okay. And
ruptured.
then every time we pierce our skin, bugs are coming in. We have bugs all over our skin. So anytime
Absolutely.
we pierce our skin, bugs are coming in.
Absolutely. And
Any
and we have to fend those off. And that's the job of the immune system is constantly, but to balance, because there's the bugs that come in and they're just in that tube, not trying to invade beyond the colon, and we tolerate those, right? So we give them safe passage most of the time. They're allowed in and out without triggering any kind of big response. And this is always, the immune system needs to recognize potential threats and also recognize friendlies, because some of these bugs are really good for us and actually work with us to help us digest our food. Um, these bugs are all very different and they're not all bad. And the immune system really needs to accurately distinguish between threats that are worth mounting a massive response that could cause a lot of damage versus good bacteria or non-threatening bacteria that are just not worth, that should just be left alone.
Okay, so now let's. So one of the people we've interviewed in the past, who gave me the description of us as being bags of bugs, argued that if we counted the number of bugs in us versus the number of cells in us, that we'd have more bugs than we have cells. Is that an accurate description in your view?
Yes, that is accurate, but they're
Okay.
tiny also. Yeah,
Well,
but by sheer number, they do outnumber our own cells. Yes.
okay. So we're going through space and time, and we are interacting with all the bugs in the world, and our system has to learn how to identify those bugs that are going to be helpful to us and those bugs that are not going to be helpful to us. And then when our system identifies those bugs that are not going to be helpful to us, our system has to mount an appropriate response to those bugs so that we are not getting injured in the process of the response. Is that a fair, very general description?
Absolutely. And I would put that also into the context of some kind of a budget that you have. You
Okay.
have some kind of total energy budget, which you can use. And you have some low budget items that you can use all the time, like keeping your skin intact. You know, there's some low budget things that you always invest in that are generally intended for this purpose. And then you have some really high budget items that you would only use very sparingly and may very well come at the cost of other things. So if you had to mount a huge response to clear something, that might mean you don't have energy to get out of bed. That all of your energy is now going to be needed to be redirected to this response. So I would think about it both in the context of what's happening and the budget with which you have to respond to this.
I love that. Okay, so now we're going to the energy portion of the response. Would if we were mounting an aggressive response against a huge threat, would that also mean we don't have the energy to respond to other threats that we've been managing in the past that may no longer have been managed by our immune system because we're using energy in another place?
Absolutely.
Okay.
Absolutely. And the system will have to triage. And so one response is going to come at the cost of another. And there's going to be, one is going to take priority over another. And in addition to that, after this response, ideally, you then want to enter into a resolution phase where you then heal from this and clean everything up and put everything back together and resolve this and do all the wound healing. That is also going to take some budget. And so if you used up all the budget on destruction and you didn't leave any budget for the cleanup crew, that's also going to be a huge problem.
Okay, so let's come back to the engineering
Yeah.
conversation and how you were defining yourself as an immunological engineer. I love that description. I wish I could have such a title. So as an and I think on some level, what I'm already learning from you is that each one of us needs to be an immunological engineer. We have to understand the system and we have to understand how we have to engineer our lives around making sure that we have our immune system functioning at as efficient a level as we possibly can. And in the spirit of you teaching all of us, not just me and Matt, but all of us on this podcast about how to become immunological engineers, let's build out how you developed this fascination as an undergrad. You then went to one of the finest graduate schools in the world, which happens to also be located in the birthplace of Lyme at Yale University. So talk to us about your time at Yale, what you studied when you were earning your PhD, and how that laid the foundation for where you are today.
Okay, great. So I feel so lucky that I got to be mentored by Akiko Iwasaki, because I got to really, as much as I studied my research topic, I also got to study how she thinks. And to this day, her voice is the voice on my scientific GPS in my head. Kind of, I don't call her up every day anymore, but she's still guiding me in some ways.
Well, she built all kinds of pathways in your brain, right? I mean,
Yeah.
she uploaded herself into you. Yeah.
Yeah, I love that. So I was working on trying to understand how a cell that is not an immune cell, how just, one of the cells of the body can recognize that it has been infected by a virus, and how it can then signal that to the immune system to trigger a response. And what became very, very interesting is I was studying one of the mechanisms by which we take out the trash, okay? And this is a really important, maintenance process inside the cell, and it's really important for healthy longevity, and so whoever in whatever household is responsible for taking out the trash, you should know you have a really important job. If you stop doing that job and trash just kind of starts to build up, that can make everybody in the house sick, right? So it's the same thing in the cell. If you stop taking out the trash and you let, things that should have been cleared just build up, what happens is you have these mitochondria in your cell, and they're really important for producing energy, but they're also really, really dangerous. And they have all these signaling molecules, and they can amplify or reduce immune responses. So they are really important, these critical points in terms of the strength of a response or the amplitude of a response. So that's what I was studying. I was studying how in the process of recognizing a virus, the mitochondria is actually a really key decision point, and the mitochondria can actually skew this decision of respond and mount an immune response, and how strong should that be, versus this cell should just, essentially signal that it needs to be cleared or even initiate a cell death.
Okay, so let's pause that for a second.
Yeah.
So your focus or your concentration during your PhD was to study cells and the mitochondria in the cells and the impact that the mitochondria plays in signaling immune response. So let's build out again.
And this process of taking out the trash, that's actually called autophagy. Yes.
Okay. So it's let's build this out a little bit. So I was always led to understand that the mitochondria is the power plant in the cell.
Yes.
That all energy is developed through the mitochondria. Can you
Not
give us
all
a
energy.
okay?
You can also ferment sugar, so you can build up lactic acid. You can do this. You have other ways of producing energy that are less efficient on a total, but much quicker. So you have choices. The mitochondria is your most efficient choice in the long term, but not the one that's always used by the immune response. Some immune cells will actually choose this fermentation pathway instead for quick, robust amounts of, energy outputs.
Bursts of energy.
Yes.
Okay.
The mitochondria can also produce bursts of energy.
Okay.
It's just, there's a different kind of efficiency and speed involved that could send you one way versus the other.
Okay. Because I want to get so you were building out this engineering process for us, and we did talk about energy a moment ago, and now we're getting to the mitochondria. So as soon as you started talking about energy, my thought process went right to mitochondria. You're not teaching me there are other sources of energy. And so what you're building out now is when you're studying at Yale and you're focusing your concentration with your mentor about cellular health. One of the things that you're studying is the toxicity that can develop in cells if there is not a cleansing process, and what impact that's having on the ability of the mitochondria to properly signal to the immune system what type of response it should be making. Am I correct in that?
Yes, I'm just going to further frame this for you, that you have immune proteins that are actually sitting on the face of the mitochondria.
Okay.
So this is not, the mitochondria needs to signal to the immune system. It's these two systems are interwoven, and they are situated such that the mitochondria is inherently signaling to the proteins that are on it, which include also immune proteins.
Okay, so would you say that based on the research you did in this as a graduate student, that you came to conclude that the mitochondria is a part of the immune system?
Yeah.
Okay.
I think that would be fair to kind of describe how I was thinking about that. Yep.
Okay. So when did you complete your graduate studies with your mentor at Yale, and where did you go from there?
2012. So the winter of 2012, I completed my PhD. And
Congratulations.
Thank you. And then in 2013, I started my postdoc with Irving Weissman at Stanford.
Okay. So what led you to Stanford? Why did you go from Yale to Stanford?
So my experience at Yale I was hooked. I was hooked. I mean, I was so fascinated. I had 1,000 questions I wanted to ask. And Akiko had just said the most wonderful thing about me at my defense, which was, she said, I usually try to encourage everybody to think outside the box, but
So when
Mikki
you say your
has,
defense, you mean you were presenting your dissertation and your mentor responded to that. Let's build this out for non-academics and
okay,
and
absolutely. So at your, at the end of, at the culmination of your PhD, you defend your research. And so you stand up in front of everybody and you give an hour-long talk about the research you've been doing over the last 6 years. And you present to your committee your thesis defense where you've written all of this up. And then afterwards they get, another hour or so to grill you on everything and then essentially award you your PhD. And so, you know, Akiko got to introduce me and say a few words, and she had said that, usually she tries to encourage people to think outside the box, but that I have no box. And I was so, so this is the state that I was in when I was, starting my postdoc. And I was just really feeling like the world was my oyster,
yeah,
and I
that,
felt
that
like I had so many questions and I had the tools to answer them. And I had, you
and
know,
you were seen by your mentor. She saw you.
yeah, and it was
That's really
wonderful.
cool.
And so I knew I wanted to have a lab of my own at some point. I knew I wanted to get there. I knew that was going to be a hard road. And what was drawing me to Stanford was Irv. I was specifically wanting to get into Irv's lab. And I saw the track record he had for turning out independent PIs. I mean, basically everybody who postdoced in his lab either got their own lab or started their own company. And it was really, just I wanted to have that opportunity. I also really liked the kinds of research questions he asked, and I saw that he was essentially asking a lot of these questions of applied immunology. He was thinking like an engineer. He was using different drugs to test what the immune system does in a way that this experimental medicine I hadn't seen a lot of in other places. And he was looking at processes of aging and how those are driven by the immune response. And I had just been working on a study on flu and aging and these cellular processes I described, autophagy and the mitochondria and how that was impacted. And so I was very, very drawn to.
Okay, so let's build this out a little bit more. So you are paid the highest compliment that anyone can be paid by their mentor. And that helped you to feel confident in taking the next step, was to go to a place where you could receive additional educational experiences that would put you in a position where you could find a pathway to a lab, where
Yeah,
you can now start to ask all the questions that people who don't have boxes can ask.
yeah,
And you would be able to also mentor students in that lab, where you can now interact with other really smart people, and you can now further develop this concept of immunological engineering that was so fascinating to you all the way back from your childhood when you witnessed, unfortunately, some traumatic events with family. So how was your experience at Stanford enhancing what you built, the foundation you had built at Yale?
yeah. So a couple of things happened at Stanford that really drove that forward. One is that I was working on this immune regulation pathway. I was working on this and I stumbled on, the bacteria that causes Lyme disease, and I started to read about it. And I started to read about the public health response to Lyme disease. And I started to read that there was always a percentage of people who never recovered, even after being diagnosed, even after being treated with doxycycline, never recovered and got more and more ill. And I saw what they were being called. I read a piece in 2007 by a very well respected scientist, and it had a box of, for the clinician, and why you would be doing your patient a disservice to even consider the notion of chronic Lyme. And I was just, to see something that had been swept under the rug for 40 years, and where, new generations of doctors were being trained to continue to just, this couldn't possibly be real. Don't even entertain this idea. I was really, that changed me. I couldn't let that go. And that's really what gave me a very strong sense of purpose of like, I need to be here. I need to be asking these questions. Part of that was actually my own feminism. Part of that was, in that, if you keep reading along that box, part of the rationale was that men are more likely to be diagnosed with Lyme, but women are more likely to develop chronic Lyme. And, the insinuation was that this is part of that argument of how this, wouldn't be real. And not, could testosterone be protective, or could, and I was just, I read that and I was thinking to myself, the fact that I'm a woman can and would be used against me if I was sick. And I was horrified by that. I mean, I'm horrified for everybody, males and females who develop this illness, and both do. And I really, I feel for everybody that has had to deal with this and has had to navigate it without, under essentially obscurity that is not unnecessary obscurity, right? Not because we can't study this, and not because we don't have the tools, but because we're choosing to actively try to ignore something that is right in front of us. And that's why I was saying earlier about stigma. I find that when a disease is stigmatized, it really hinders the kind of questions that we're asking in science, and it creates an opportunity for those who are willing to.
Well, let's come back to the box metaphor, right? Yeah. I mean, your mentor described you as somebody who doesn't acknowledge boxes, right? Mm-hmm. And really, what was happening with that article you had written, putting aside the gender piece, which I'd like to build out with you in a moment, is he was building a box, and he said, right, all of you stay in that box. And of course, the Tal response is, but boxes don't exist. I don't acknowledge boxes, and you cannot, in your little side note, and your message to clinicians, signal to all of us that we have to stay in the box that you're building for us because I don't have boxes, right? And to
maybe I saw it as a challenge.
And to make it even worse, what you're doing is you're creating a gender box, right? And I'm certainly not going to live in the gender box because that's something that I find troubling. So the PhD without boxes now has a challenge, right? She's being challenged by what is really just box building or stigmatizing a certain disease and the way that people are going to be treated with disease.
Yes. I think the other major thing that happened, and this would then evolve over the, over the pandemic and over the adaptations that we were all making to connect in virtual space, is that I used to say that the best, in my early postdoc years, I used to say that the best part of my job was that I got to wake up in the morning and ask, what are the most important questions I could ask? And I loved that. I'm so curious, and I just loved this opportunity to answer my own curiosity.
So
what happened,
I'm sorry.
But what happened is that I realized at some point as I started to work on chronic Lyme that was too big a responsibility for somebody who had never had chronic Lyme, and that I needed to be talking to patients. And I think that's the other big change that happened in me over the pandemic, but also just during my postdoc, and was something where I was really fighting up against, the objectivity of science, and you keep yourself objective, and people were looking at this as a liability. How,
Looking at what
how
is
much I was
liability.
engaging with the patient
Oh,
community.
okay.
But
So,
I found that to be really important. Yeah.
yeah, I know. I mean, and obviously a patient podcast like the boot camp is gonna very much agree with that perspective, and we're gonna build that out. So let's stay with the foundation because Matt is dying over there because he
Rich, you're
hasn't been
killing
able
me,
to
Rich. You're
join us.
killing
I
me.
know, but let me build out the foundation for those of us who are of limited scientific capacity. So you're an undergrad. You're learning about the immune system generally, learning about cellular processes. You made the observation that the mitochondria is now a part of the immune system, and it's constantly communicating with the immune system, not just signaling, but constantly communicating. So there's an ongoing conversation between the mitochondria in your cells and the immune system generally. Um, and now you're now, I guess, as part of the enrichment that you're getting at Stanford, now learning about how these microbes are interfering with the communication between the mitochondria and the immune system generally. Is that accurate?
Yeah, that's accurate.
Okay. So
You get an A.
all right, thank you. I'm glad I am the runt of the podcast. Matt is generally much speaking at much higher level, but I always try
That's
to
not true.
break it down so that I can understand it. And I think folks, again, certainly new to the community want to understand these concepts. So now you're now focusing on a particular bacteria, right? A
Yeah.
bacteria that was all around you during your childhood, at least during the first 6 years of your life when you were living in Connecticut. Certainly all around you while you were a graduate student at Yale, and now you're on the West Coast.
Mhm.
A place, by the way, who many argue there is no such thing as Lyme disease, which is always ironic to us, right? And now you're introduced to this corkscrew bacteria, and you're studying the impact that this bacteria is having on the mitochondrial communication with the immune system. So help us how that became the next part of your passion, and how that's now taking you to the place where you are now at MIT.
Okay, great. So, I'm looking at how I was studying how this bacteria manages to convince the immune system to ignore it. And this is a really critical process for any chronic infection. So any chronic infection has to be able to do this. Any cancer has to be able to do this. So there are things that would normally be cleared by the immune system. And if it is able to persist, it has managed to fool the immune system into leaving it alone. And so I was studying this process by which this particular bacteria, Borrelia burgdorferi, is tricking the immune system into leaving it alone and just like, everything's fine, don't worry about me,
I'm
I
not
should
here.
be here. So I was studying that process. I was studying that how that the mechanisms of how that works, how the bacteria is signaling to the immune system. And what I was really getting very fascinated with is if we could change how we try to predict illness trajectories. So I started to become fascinated with this question of
What do you mean
how
by that?
early.
What do you mean by illness trajectory?
Yeah. So you could have an infection like Lyme, and you could get antibiotics and you could recover and never think about it again. Like, oh, did I have Lyme? Oh, yeah, maybe, like this was a non-event, right? Versus you could have Lyme, you get antibiotics, not recover. This could change your life. You could now go down a slide of worsening, and your life is never the same as before you had Lyme. These are different trajectories that you may now go on after having had this infection.
Okay,
And
can I ask you to pause there for 1 second?
yeah,
Because you gave two scenarios based on one input. And I want
Mhm,
to focus on this from an engineering standpoint,
mhm.
right? So can you first describe what are the two types of immune responses? We have an innate immune, or is there more than two?
There's there's more
Okay.
than two.
All right. But at least the way we've been taught it, there
Okay.
is the innate immune response, right?
Yes. Mhm.
And what is the other response, or are there more? All
The adaptive.
right. So we have
So
innate or adaptive, right?
exactly. So if you want to put it in this framework, you have cells that are part of your innate immune system, and they
Yep.
are always on, essentially, and always available, and they have receptors on them that have been given to you by your ancestors who survived an infection, and they had ways of, you know, of proteins on these cells that could recognize different infections, trigger a response, and you would survive that response. And you have inherited from your ancestors the recipe to make those proteins on your innate immune cells, and your innate immune cells are going to respond in this coded way that you have inherited. Okay, so
Okay.
this is one branch of the immune system.
So the software for responding to a particular type of microbe
You
was
were born with.
was was born. Okay.
Yes. The other branch, the adaptive immune system, has, B cells and T cells that are combinatorial. So there's an
What is
element
it?
of chance here that you are not necessarily born with, but that any given thing that you see that was not there when you were a fetus, and so something that your body doesn't recognize as self is something new that's being recognized. There is a chance that you could recognize that and respond to it, and you intentionally create the proteins on these cells in a way that could recognize an enormous variety of different new things. And what you do is you
New
train that.
threats.
What's that?
New threats.
New threats
Okay.
and changes in your body, and you train that on your body constantly to make sure that they're not going to attack you. The goal is for them to not attack you, but only attack things that aren't you.
Okay, so let's come back to Borrelia. So you gave the hypothetical of somebody has Borrelia introduced into their system, and one person would take doxy,
Yeah.
a broad spectrum antibiotic, and the Borrelia would not affect them for the rest of their lives. It would be cleared. And another
Yep.
person would have a different response despite taking the antibiotic. Let me ask you another scenario in the same vein, which is I get bitten by a tick. The Borrelia
Mhm.
is introduced into my system. I don't get sick, even though I don't take
Yep.
doxy.
Yep.
Matt gets bitten by a tick.
Yep.
He does not take doxy either, and he becomes chronically ill. So
Yep.
can we focus on that piece as well
Yep.
as the
I'd
introduction
love
of
to.
the now, because I would, and correct me if I'm wrong, I'm assuming that doxycycline is a tool for engineering the immune response. So when we footnote that or bookmark that for a second, and let's
Mhm.
focus on the
Bit by a tick, get sick, don't get sick. So let's
yes,
let's start with that. So
and no doxy, right?
one possibility is the tick didn't have Borrelia in it,
Okay.
right? So you could have both been bitten by a tick, but somebody got lucky and somebody didn't get lucky. So that's option one.
Yep.
Now let's talk about option two. You
Mhm.
both got bitten by a tick that had the exact same Borrelia burgdorferi in it,
Same
same
strain.
strain. So
Okay.
it's not that one is much more aggressive than the other, or we're talking the same strain, but two people mount a completely different response.
Yep.
So this is where we get into what I'm very interested in of these illness trajectories. Which way are you headed, right?
Perfect.
And one thing that we know is that Lyme has been around a lot longer than we have been diagnosing it. And a lot of people don't show at least the classic symptoms that we're looking for. So they don't get that big bullseye rash or the swollen knee or ankle or the carditis, they carry it kind of silently. Now, it's a persistent infection in mammals. So if it has ways of tricking your immune system into ignoring it. It can do that in mice, it can do that in rats, it can do that in humans. So if you don't get antibiotics that can completely kill it, you will stay infected. This isn't an infection that your immune system has the ability to clear on its own. It cannot. However, let's bookmark that one because because there's a major footnote on that. Okay. But in general, once infection is established, and that's what we'll come back to, were you able to prevent infection from getting established? But if you did not, if infection is established, as having a mammalian immune system, you probably do not have a way to clear this infection, and it will stay established. But the way in which your immune system responds to it could be very, very different. And this could make the difference between an asymptomatic infection, means you have the infection and no symptoms that have been attributed to Lyme, or you have the infection, your immune system sees it, your immune system is attacking it, your immune system is continuing to attack it, your immune system will take you down with it. And it is literally mounting a response that is going to burn the house down, and you will just fall apart over the response to this infection. And so, these are two very different illness trajectories, and they happen in people who could have seen that the same bacteria, but a difference in how their immune system responded could have meant the difference between quietly ignoring it with all the other bugs that we have in our body and that we are, continuing to function, versus, putting everything onto, eliminating this bug.
So can we define infection? Does infection necessarily lead to disease, or is infection just your body's harboring this hostile bug?
This is a fantastic question. So I guess what we can do is we can use the term colonized in the context of, a truly asymptomatic response. The bug is causing no damage, your immune system doesn't mind it. This we can call colonized, versus infected where there's a problem.
Okay. So now one of our favorite guests, I'm sorry, go ahead.
Okay, I want to circle back to my footnote.
Okay.
So we did publish a paper together with Hannah Olila's group on this secreted protein. And now the name is a mouthful. Okay, so I'm warning you in advance. SCGB1D2. I did not name this thing. And we are trying to call it line block. It's a secreted protein. It is in your sweat. It is also in other secretions. And the reason we stumbled on this protein is that Hannah had done an analysis that's called the GWAS, where she looks across genomes of many, many, many people. So across these recipes of DNA that they've inherited from their ancestors. And what she found is that people who were diagnosed with Lyme disease more often had a mutation in this protein than people who had never been diagnosed with Lyme disease.
Really? Oh, that's
and
fascinating.
So I did want to give you that footnote, then my lab studied this protein, and what we found is that it could prevent Borrelia from growing. And if we gave it to mice at the same time that we gave them the infection, it could prevent infection.
Wow.
So that's why I was saying you do have ways to prevent establishment of infection, and we do have, as a population, differences in this protein that can be part of that difference. Did you get the tick bite? The tick had the bacteria, but you were sweating and you had all of this protein and you were able to just, versus wow. You either have a mutation in this protein or you weren't sweating, or there was a difference there that resulted in you not clearing it on contact, and it managed to establish infection.
So I'm supposed to take you to your lab, and because Matt is going to go crazy, but I have to.
No, this is too good, Rich. You can't move on. I'm with you. This is too good.
The this footnote that we just focus on,
Yeah.
I am
Mhm.
so fascinated by this. So one of our favorite guests is Dr. Bill Rolls. You may have met Dr. Rolls at the Fashion Week. He was also a model like you, and one of the things that Dr. Rolls argued in his book Unlocking Lyme is that humans have been getting bitten by ticks as long as there are humans.
Yes.
And that, of course, the bacteria, Borrelia, actually is older than we are, right?
Yeah.
It has existed longer than humans have existed. Um, and I'm just fascinated by the prospect of different types of immune responses to this bug that we've been coming in contact with. When I say bug, I don't mean the tick. I mean the
Bacteria.
Borrelia, right? The bacteria
Yes.
Borrelia, right? And as part of our adaptive response, I mean our adaptive immune system, of course, because we've been getting bitten by ticks for as long as there have been humans, and because we've had this bacteria injected into us for all of time, there are various immune responses that should be able to, I would argue, clear this infection without the introduction of engineering. Now, one of the immune responses, which I never heard of until just now, was that in our sweat, so of course a tick is going to be biting us, right? And
Mhm.
it's going to be injecting its mouth part into our body. And one of the things and of course, it's piercing our, the largest part of our immune system, which is our skin, right? Or I should say our protective
Mhm.
system. As it
Absolutely.
as it's injecting into as it's biting us, we're now our sweat glands are now pumping
Mhm.
sweat into the area where we're being bitten,
Mhm.
and there is an immune response
Mhm.
and a protein in that sweat that is now protecting us from getting Lyme.
Yes.
That's fascinating. It's just a brilliant immuno response, an adaptive response. And
And
you
what we
discovered it.
found, yeah, and what we found is that the mutation that is more common in people who've been diagnosed with Lyme, that if we compare that mutation head to head with the reference version, that what we see is you need twice as much protein of the mutated protein to have the same effect in
Wow.
terms of preventing Borrelia from replicating and from growing. So it still works, but it works only half as well.
And of course, because it works half as well. Your immune response is going to be muted.
That could make the key difference in terms of, did that tick leave you with 200 bacteria and maybe you had just enough protein to attack 200 bacteria, or did that tick leave you with 200 bacteria, but now you only have enough protein to attack 100 of them.
Wow.
And
I-
so 100 managed to
Look,
get
it's
through.
it's it's really so in the spirit of the engineering conversation. One of the things that Matt and I discovered when we were doing research on building a tick kit is that there was a study out of Vienna. I don't remember, Matt, you may remember, a study out of Vienna that indicated that um, if you applied Zithromax Zithromax cream on the site where a tick had bitten you within 24 hours of the bite, that you would substantially reduce the likelihood of an infection. So I'm just wondering whether or not that is essentially providing a similar response as our sweat glands would be providing in the proteins in the sweat glands when a tick is biting us.
I think so. I very much do think so. And this is where, I'm saying there's so much low-hanging fruit. There's so many kind of great options for us to engineer. We can control what you do immediately after the tick bite. There's so many things that we could change. So it's not just, doxy is our only option, and you either, it's doxy or nothing. There are so many more opportunities for us to consider to supplement doxy in ways that ideally everybody could recover.
So another thought that I have is when our skin is full of bugs, right? I mean,
Yeah.
our skin is protecting us from these bugs getting into our system. And when the tick pierces our skin and I'm assuming a lot of other bugs are going in addition to whatever the tick is spitting into us, which of course is going to mount an aggressive immune response. And I'm assuming that what you had discovered, we're gonna have to call this like the tau protein now. The tau
Oh,
protein that
it is
comes.
now the tau protein. It has
No,
to
no,
be. That's it.
we have nicknamed this Lyme block.
All right,
And
that's all right.
Lyme block, aka the tau protein. How about both? We can do both.
So when the tau protein comes gushing over the site of the tick bite, I'm assuming it's also going to be killing other bugs that are coming into the system at the same time that the bugs that are inside of the tick's gut are being spit into you.
You have now addressed one of my questions for this, is we have 11 of these secretoglobins. So
Secretoglobins are called?
yes, so that SCGB1D2, the SCGB is for secretoglobin.
Okay.
Yes, did not name this, but we have 11 of them. And they are expressed in different parts of our body. And most of them are actually coating our lungs. And so I very much have this question of, do you have different secretoglobins that attack different bacteria and viruses and fungi? Are these part of our antimicrobial defense that we have not been studying? And I think there is some rationale to support that. Our research, but also others on some of these other proteins. And I think it's a very important area of research.
You're melting our brain right now. And I'm glad we renamed it the tau protein. So, holy cow. All right. So let's come to the, I promise, Matt, let's come to
Rich, come on.
let's come to the lab. I mean, Matt, look, I can't help that my brain keeps getting melted with all of these different cool questions that Dr. Tala is causing me to ask. So let's move to your lab so Matt can finally join us. So
yes.
So
we're at MIT now.
You finished your work at Stanford and all these brilliant things that you're why MIT and what is the foundation of the Tau lab? Okay,
so why MIT? We're going to go back to that gender question from earlier for a moment. So now you're with me at a big experiment endpoint at Stanford. I've got my entire team working on this. Is an all hands on deck day. There's people down in the mouse room. There's people up in the lab. Everybody's processing tissues. The people who go down to the mouse room go down with these little plates with 24 little wells to collect these little mouse uteruses from an experiment that we called male versus female. Okay. The name is fairly self-explanatory. Everybody's getting infected and there's all these other conditions happening. And the question here is about the male versus the female response. So for the females, we're collecting uteruses, and one of these animals is really chunky, and I'm commenting about that because I love chunky mice. Love it. I love baby fat rolls. I love chunky mice. Like, give me a big fat OB. I'm like, oh my God, the cutest thing, right? So I might have commented, about this thick little mouse. And when we opened her up, she was all uterus. This uterus was enormous. It was full of Borrelia, it was so swollen. This thing looked so horrific. I never got it out of my head. So you're lucky, because when I give talks, I show it. And I'm like, this mouse changed my life. Let's see what it does for you. And then everybody loses their lunch. But I actually called up Linda Griffith at MIT. She's the director for the MIT Center for Gynepathology Research. And I said, do you want to see something gross? And she said, always. And I showed her this animal, and she was like, we got to figure out how to get you to MIT. Wow. And she did figure out how to get me to MIT. And we have been really just having an incredible time asking really big questions ever since. She has changed things for our understanding of endometriosis and other gynecological diseases. And so we have a paper out together led by one of the researchers in my lab, Paige Hansen-Colburn, that is all about how Lyme disease increases risk for multiple different gynecological conditions.
Wow.
So Linda got me to MIT. She made me the associate scientific director for the Center for Gynepathology Research. And that is how I'm now at MIT, have my lab. So now we're here.
Okay. So quick question, Dr. Tal. Well, this is definitely not a quick question. I don't know why I'd say that. But when it comes to gynecological health and it comes to Lyme disease, Rich and I, right off the bat 5 years ago, when we started Tick Boot Camp, we realized that the female population in the Lyme community, which seems to be pretty dominant. I think I'm the minority by far as a male who is severely impacted by chronic Lyme disease. But the female population in the chronic Lyme community, almost all of them suffer from severe endometriosis and female reproductive system problems with chronic Lyme. Some of them have had full-on surgeries to remove all their reproductive organs, and it still didn't have much success. But we know there's definitely a connection here. So this is a huge, huge topic on our podcast. So can you help us go a little bit deeper? We want to learn, why does Lyme disease seem to just get attracted to reproductive organs for females and cause this severe, severe inflammation and pain in the woman population?
Yes. And let me just preface that we are working on a follow-up study, and I have a postdoc, Guido Pisani, who is working on this, but we have also looked in males, and we also find Borrelia in the male reproductive organs. So this is not unique to the female reproductive tract. So I just want to put that out there. But the extent of damage to the female reproductive tract, that is what we found really shocking. And what we find very concerning is working together with NASA Sina Armstrong at Fred Hutch, who did this, exploration of epidemiological data in humans. They saw that after having Lyme, even after having doxycycline, there's about a twofold increased risk for many different gynecological diseases in humans. So there was this connection there that we were really shocked to uncover, because it hadn't been, very well studied in the literature before that. But what to me was the biggest surprise is that we knew very well that Borrelia loves the bladder. So in every mouse model, everybody sees always infected bladders. In humans, there's, one of the tests that was, meant to be this novel new test looking in the urine for antigens of Borrelia. So, this is something we understood very well. Borrelia colonizes the bladder, and that's in males and females. Now, in females, there are two very easy ways to get from the bladder up the vaginal canal into the uterus. And to me, I think what was most shocking was not that Borrelia was in the female reproductive tract, but that people hadn't been looking for it there. Because what we know from UTIs is that if you have a UTI in your bladder, very likely that is also making its way up the vaginal canal, and that can also be found in the uterus. And so if we know it likes to colonize the bladder, at least, then we need to check what's happening in the uterus, because it's quite likely that we will find it in both places.
So Dr. Tal, it seems to make sense to me. I have a whole bunch of questions that I want to follow up, but I don't want Matt to feel excluded even more.
Nope. Rich, I don't mind, as long as we're thinking the same questions. Even when you were talking, I had the same questions you had. So
it would make sense to me that Borrelia would want to colonize in the bladder and get to the female reproductive system, because you would think that Borrelia wants to move from host to host. And one of the fastest ways of moving to another host, short of another tick bite, taking the Borrelia out and biting another being, would be to go into the female reproductive system, because that's where there are going to be more hosts developing, and they could go to a new host and then continue.
Let's roll that back. So if we're going to talk about what Borrelia wants, let's discuss the fact that ticks can't pass Borrelia to their offspring. So Borrelia can't be transferred through that egg stage. And in ticks, unlike in us, we think that Borrelia is a beneficial infection, or at least, well, okay, a beneficial microbe, or at least a, symbiote. So it at least causes no harm, and it very well might be helping. And there's a
No,
couple.
it does help, but it ticks who have Borrelia have a higher fat content. They survive longer. They can survive in cold weather. So it certainly has a positive
And they may
relationship.
feed faster. And feeding is the most dangerous time of their life,
Okay,
right? They're on the mammal, one scratch and they're dead, right? So
right.
being able to fall off faster. So these are reasons why we think this is, helping them survive. We think this is a beneficial
For sure.
microbe for them. So the problem, though, is that ticks can't pass it to their offspring. So Borrelia has got to ride out the winter in a mammal that can reinfect a new crop of ticks in the spring, right? So this is probably one of the main drivers of sustaining Borrelia infections in these small rodent populations. That the larvae, that the larvae or nymph ticks can bite. One thing that, we don't know, we really don't know to what extent vertical transmission, so transmission from, mother to child in mammals, could be sustaining this infection. We don't know that. But the ecology that we know is ticks are biting the mammals, and then in the spring, a new crop of ticks, are biting these mammals, and then the summer and the fall, and so that's going to maintain this transmission of the microbe by riding out the winter in a mammal.
Okay. So let's talk about the difference between the male and the female immune system.
Okay.
And this is one of the things that Matt and I have been focusing on from the very beginning. And there are folks who are accusing us of only reaching out to women to interview on our podcast, which was ridiculous. We were reaching out to people who were sharing their experiences largely on social media, and we were asking them to share their stories, the challenges and their successes. But our podcast was largely a female podcast, at least from the audience standpoint. LymeDisease.org then finally did a study based on the 10,000 people who had answered their questionnaire, and I think Lorraine Johnson and others had published a piece indicating that Lyme is affecting women differently than men.
Yeah.
So can you give us some insight into the difference between the male and female immune system, and why the Borrelia may increase the likelihood of a chronic illness in a woman at a greater rate than it would a man? By contrast, and give me your thoughts on, it seemed like the female immune system was better at managing COVID than the male immune system. So are the distinctions in
let's break that down.
Please do.
So when I got to MIT, one of the first things I did is started a seminar series called Sex and Immunity. And that is because this has been a big question of mine as well. Why, what is the difference between the male and female immune response? And I have been very, very inspired by the different researchers in the field who really have been pioneering this study. And I think, one thing I would want to start off with is, since you brought up the difference between COVID, actually the first keynote speaker we invited to launch Sex Immunity was Sabra Klein, because she's been one of these real pioneers looking at the differences between the male and female immune response. And she studied this in the context of both flu and COVID. And with flu and COVID, what you see is that age-matched, males are more likely to die from acute infection. But if they survive it, they are much less likely to develop chronic illness from it. Females, on the other hand, are more likely to have a mild infection in that original infection, but then also more likely to go on to develop chronic illness. So we're going to footnote that and come back to it. because the next thing I want to say is that every immune cell has receptors for steroid hormones that are different between males and females: testosterone, estrogen, and progesterone. So every immune cell has receptors for these sex hormones, and they impact the function of these immune cells in ways that we don't understand. And this has not been sufficiently studied. And this is one of the big things that Linda Griffith and I are really, this is one of the things we're constantly talking about and plotting and planning of how to study this well. And this is a huge focus of mine moving forward, is this systematic study of what really drives these differences, and to what extent are these differences getting driven by different levels of these hormones or fluctuations of these hormones. But okay, circling back to women being more likely to develop chronic illness, one thing that I have asked myself a lot, and I would like to expand women to caregivers in this context, is I want you to think in a historical context, that you were going to have people who are out, if we want to think about hunters gatherers, or if we want to think about cave times, or we want to think, you had people who needed to be able to run at full speed, right? And so it would make sense for them to be 3 days knocked out cold, all energy diverted to clearing this infection. They can't move, think full-blown man flu, right? And then on day 4, be back up to full speed. Versus somebody had to be caring for the young. Somebody had to be making sure that people were fed and stayed alive, and being just knocked out for 3 days would not have been an appropriate response that would have kept the species alive if everybody did that, right? So I think there must have been a balance where some people responded this way, of just divert all energy, and some people respond with, okay, let's, divert some energy to that, but let's still make dinner, and let's still change diapers, and let's still make sure that nobody's, running over cliffs or anything. And I think that balance in the population has also led to this balance that we see now in the differences between an overwhelming response to acute infection versus this risk or vulnerability to developing chronic illness.
Okay. So let's talk about post-infectious responses. Do you believe there is a post-infectious response, and which may be causing the chronic illness, or do you believe it is only infectious?
I don't like the term post-infectious, not because I don't believe in it, because it's making an assumption that I don't know if we can make. So I try to use terms that are more descriptive. So I mostly try to stick to infection-associated chronic illness. And if I say Borrelia infection-associated chronic illness, we know every piece of that. You had a Borrelia infection, you now have an associated chronic illness, that comes from that Borrelia infection. Is the infection still there? I don't know. Is this now an immune response gone rogue and it's trying to kill you? I don't know. There's a lot of my research is very humbly admitting that I have no idea what's going on, but I just want to measure everything I can possibly measure. So MAESTRO, we joke that the acronym for MAESTRO is measure absolutely everything so that we can have reproducible outcomes and or reliable outcomes. And that's really, really the goal. And that's so much of what we do is, just measure everything. Let's write a new textbook for the, medical schools that, instead of sweeping this under the rug, let's tell them how to measure it. Let's tell them what it looks like. Let's make a new specialty that is focused around these illnesses and that knows what they're looking for and what they look like.
So one of the things I want to just put on your radar is that as you've now learned, we've renamed a protein a tau protein. So now we also have a new definition. We have a different definition of Lyme disease, right? We don't define Lyme disease the way the CDC has defined it. And quite frankly, there seem to be as many definitions of Lyme disease as there are researchers and doctors. So we define Lyme disease here at Tick Boot Camp as a polymicrobial, multisystemic, chronic, infectious, or post-infectious disease. So the reason I'm now calling it to your attention is because you were sharing with us that in one of the footnotes that women who are who have a reduced response to the acute infection in many cases will have a more severe chronic presentation, and vice versa with men. So that's why I was bringing you to the post-infectious piece of our definition. My antenna went up as soon as you shared that research finding with us about whether or not it really is a post-infectious illness as opposed to an infectious illness or an infection-driven illness.
And that's one of our big questions. It's a huge question. And one thing that I would say, is that one piece that we do consider within that is that after sexual maturity, women are making immune responses for two, and we know that there's antibodies in the milk. There's all sorts of ways in which we deliver, these we share our immune responses, and so we make more profound responses in general. And so it's that's probably part of it. I mean, that's definitely involved. There are aspects of the immune system that are meant to protect not only the woman, but also her children.
Right. So but you talked about stops. You said that there are different stops, meaning the immune system is going to stop at different times. In fact, it was the foundation of your early research, right?
The checkpoints. Yep.
The checkpoints. So is a checkpoint a stop where your immune system stops responding?
It's a stop. Yep.
So you're leading me right to the question I've wanted to ask everyone and nobody's ever been able to give me an answer to, which is it possible that Borrelia is tricking a stop that is affecting women at a greater rate because the female immune system has greater stops in order to be able to protect the baby that is developing in the woman, and there has to be the capacity to protect this foreign body that is forming inside of a woman. So is Borrelia possibly tricking a stop that is affecting women differently than men, which is why we are seeing more women chronically ill from Lyme disease rather than men?
So I love that question. And I'm going to afterwards give you guys several links to link in the comments of different publications that we have. One of which is this study on this checkpoint where the Borrelia has this protein called P66 and it interacts with our checkpoint. So we have this negative regulator that's a stop. So this stop is called SERP alpha. And
I'm sorry, it's called what?
SERP alpha.
SERP alpha.
Yes. And I always love to play around with surprises of SERP alpha. So SERP alpha is a stop. And so this is this major checkpoint. And is it different in women versus men? And is it different because of, needing to be able to tolerate, this baby that's half dad? I think that's an extremely interesting question. I would love to, go in and check the differential expression of SERP alpha in females and males. And I can't believe I haven't done it. My goodness. I very much should. So great question. For part one, Borrelia is manipulating these stops. For part two, these stops are different in males and females, specifically ones that are, local to the uterus and mediated by NK cells, particular types of immune cells that really survey the fetus and shape the immune environment of the uterus. Could the same stops that Borrelia manipulates be differentially expressed in males and females? Absolutely. And that's a really important question. I love it, and love to come back to you on that, after I've had some more time to think about it and look into it.
We definitely gonna have you back because Rich is
Answerable.
hogging you, Dr. Tal. And I'm not, so we're gonna have you back again. To be fair, I stole you at the fashion week, so I had you, but now
and you are Matt's birthday present, so I will let
I know
Matt.
you're ruining my birthday, Rich. It's not really my birthday. So let's talk about Maestro, because I think it's really cool, and we've touched on it, but we didn't really geek out about it too much yet. So can you first just reintroduce what it is again, but then tell us what technology you're using, because you have so many patients that are coming in this largest clinical study ever in MIT, and using cutting edge tech to help identify patterns and trends and treatment modalities for the chronic Lyme community.
Yeah. So the idea behind Maestro is literally measure absolutely everything. That's the concept. And the idea is that patients can tell you something's wrong. And they feel that. And there's a lot of surveys that people answer, and the survey shows that something is clearly wrong. But then, they go to their doctor and they say, I'm feeling brain fog. And the doctor's like, okay, I don't have any way to measure that. And even if this doctor believes the patient and is, they're willing to take this survey, 0 to 10 at face value and use that to mean something, that's not going to unlock a diagnosis. That's not going to unlock insurance reimbursement. That's not going to unlock clinical trials and new treatments. And what we wanted to do is listen to our patients and participants and take all of this patient reported outcome, all these survey data, and match it to everything and anything we could think of to measure, and then ask which measurements fit with which survey data, so that in the future, when people are saying, I feel XYZ, the doctor can order, ABC lab test and actually see something, not just the complete blood count. We know that's not going to show anything, or, this is more complicated than just anemia, or just a vitamin B12 deficiency, or just a vitamin D deficiency. This isn't something that people just have access to measuring and fixing. And so the idea here was, let's measure your brain waves. Let's measure your eye tracking. Let's measure how your eyes move. When we ask you to make a circle, what are your eyes actually doing? Can you make that circle? And let's look just underneath your nail bed at those smallest little blood vessels that you have in there called capillaries. And let's look at the blood that's going through the capillaries, your red blood cells, your white blood cells. What kind of formations do they make? And how does that look before and after you do something we call the Nasaline test? So what's the Nasaline test? This is where you lie down for 10 minutes and then you lean against the wall for 10 minutes with your feet on the floor and your back up against the wall. And not everybody in our study can even complete this, because they're about to pass out during this 10 minutes on the lean. And what we do is we take these pictures of their capillaries right underneath their nail beds, before and after the Nasaline, on their fingers and their toes. And we're seeing really profound stuff, and this is painless. This is a picture.
What kind of stuff? You got to give us a little bit more. What kind of cool stuff are you seeing?
Oh, so some people have no symptoms on the Nasaline. So I've tried this myself. I don't, I'm lying down, then I'm leaning. People like me, you don't see any difference in my capillaries before and after I do this. However, people who are about to pass out, people who are feeling massive symptoms on the Nasaline, they have profound changes in this microvasculature before and after. And so we can see things like enlargement of the capillaries, so they're now just loaded with blood, or we can see things that look like the blood is in the wrong places, it's not being held right, We can see tissue that looks like it's getting starved of blood. There's almost no capillaries there. So there's these dynamic changes that are happening that we wouldn't expect this process to be so dynamic. We would expect something more fixed. And so the fact that it's this dynamic points us towards something that's wrong and gives us something that we can measure. And so, that's very powerful. We have, my gosh, so many different things that we're measuring. So our participants have been incredible. And until we added in the EV rings, there was no compensation. And even now, the EV rings are the only compensation that we give. And yet people are giving us, throat swabs, vaginal swabs, rectal swabs, urine, saliva, sweat. I mean, just everything, every biological sample. They're giving us blood, lots of it, so that we can measure all of these different proteins, lipids, metabolites, we're doing other things like we're looking at, are they hypermobile or not hypermobile? And there's reasons that we're looking at that.
Dr. Tal, the hypermobile is really like Ehlers-Danlos syndrome, correct? It's sort of like you're hyperflexible, which is something we've observed too on Tick Boot Camp. Again, very early on, we didn't even know what EDS was, Ehlers-Danlos syndrome, hypermobility. But it seems that a very large percentage of the chronic Lyme community is hypermobile. Why do you believe that people that are hypermobile are more prone to chronic Lyme?
Yeah, so one of the researchers on my team, Beth Pollock, actually had this question right from the start, and we started looking into it in our mouse model. So in our mouse model, what we do, as opposed to the clinical study, in the mouse model, we're comparing two different strains of mice, Black 6 and C3H, and they respond in very different ways. So you asked me at the beginning, two people get a tick bite, two people get the bacteria, they have different responses. We get to study that in the Black 6 versus C3H. So we can infect them with the exact same bacteria, same amount, and then watch how their response is very, very different. And the Black 6, they kind of control it. They have a very macrophage heavy response. They keep the bacteria at bay. They never clear it, but they keep it at bay. They do get terrible gynepathologies. So I'm not going to say they tolerate it, but they don't fall apart. The C3H mice are falling apart. So the Borrelia is in the collagen. These mast cells are coming to the collagen to attack the Borrelia. They're attacking the collagen. This is happening in between the vertebra, all along the spine, all along their mouse tails where these vertebrae go throughout the tail. And what we see is that the attack against the bacteria is just really compromising the structural integrity of these animals. And so in our clinical study in the humans, we're asking about this, we're looking at this because in the mouse model, we can show a strain of mice that do not have genetic EDS. There is no genetic defect in their collagen genes. They are wild type mice. And if we don't infect them, this doesn't happen. But if we infect them, this does happen. And specifically in the C3H versus the Black 6. And so we're studying that in the human system as well, so that we can say to what extent is hypermobility either a pre-existing risk or something that develops after the infection because of mast cells, because of compromising the collagen integrity where the bacteria are hiding.
So to kind of repeat or rephrase that one part is, because we hear this a lot, we've had people like challenge us on this podcast. I have genes that say I'm going to get Ehlers-Danlos syndrome. It's not tied to Lyme. And I think you're saying, yes, some people have that gene and maybe they would have gotten it otherwise, but people that don't have that gene in mass are still getting hypermobile conditions without the gene, which is now a indication that Lyme is bringing it on because the bacteria is essentially attacking all of the collagen in the connective tissue. Is that a fair assessment?
Yes, except not all the collagen.
Okay,
but yes.
Some.
Yes.
Why not all? Is that just meaning like it's just going through the body and attacking what it can, or is it looking for certain types of collagen?
Every fiber of collagen in your body has Borrelia with it. It's the Borrelia are in certain types of connective tissue, and not others. And so not all, because it's in specific places.
Got it.
You have different types of collagen, different types of connective tissue. Yeah.
Does that same rule apply to other illnesses, right? Because Lyme is known as a great imitator, especially with your world, with immunology. There's a ton of autoimmune diagnoses that come and people say, I definitively have these 7 autoimmune diseases. But is it really that they had the genetic expression for that, or that the Lyme bacteria activated the immune system, as you call it, in a catastrophic way, mimicking or essentially turning into these autoimmune diseases, but it's not the true genetic flavor of that autoimmune disease? Can you break that down a bit? Because I don't know that I said that well.
Sure. So I'll tell you something that we see. So we have worked together with the Lyme Biobank to study their samples and what kind of immune responses are developed. And I'm going to now circle back to something I wanted to tell you guys about how we study these illness trajectories. So a big part of my lab is trying to ask this question about illness trajectories. So how do we know from that initial infection which way you're headed? And this is a really important question in my mind, because when I was asking around why we've never done a clinical trial for chronic Lyme, why we have no treatments for chronic Lyme, what I was told by industry is if you have a disease that 90% of people are going to recover and 10% of people are not going to recover, and now you're testing a drug and that drug helps half of those 10%. So 5% of people now recover who wouldn't have otherwise recovered. You're telling me I'm going to run a clinical trial that's going to be enormous because there's no gold standard assay. So I'm going to have to give doctors, any test they want to run to make this diagnosis. And then I'm going to be looking for the difference between 90% recovery and 95% recovery. They were telling me this study would be huge. It would be an enormous trial and nobody's willing to take on that cost. They told me, you tell me who the 10% are. I'll try a drug tomorrow. I could have, they said, if you tell me the 10%, if you can predict them before they develop chronic Lyme, if you can predict them from infection, now I can take 20 people in a doxycycline only arm, and you're telling me they're not going to recover, and I can compare them to 20 people that I'm testing this interventional treatment, and now, half of them recover. I have a signal. Now I can move that on to the next phase. And so I took that to heart, and I said, okay, so what I need to get you is a prediction of illness trajectories. I need to be able to ask the immune system, are you clearing this infection? Are you mounting a protective response? Or are you going to bring the house down? And that's the question. And so the question we have been asking the immune system up until now, when we do diagnostic serology, so when you get that blood test, looking for antibodies for Lyme, we're literally counting IgG antibodies, so one type of antibody, against one or multiple proteins on the Borrelia. The question I'm trying to ask the immune system is an open-ended question. So move beyond this yes, no, you have seen this bug or not, but asking the immune system, if you saw this bug, how did you respond? And that now opens us up to this ability to not only diagnose chronic Lyme, but to diagnose it before it happens. So to say you are at very high risk of things going very badly from here. So now let's focus on this subset of people, because the other people, we talked about them, they're going to get Lyme, they're going to get antibiotics, they're never going to think about this again. That's a lot of people. Right? And that kind of makes it a very complicated thing to study. And if we can focus on the people who are not on track to recover, whose immune system is not making a protective response, who are not clearing the infection, who are not responding in a way that is going to be sustainable, if we can focus on those people, everything changes in my mind.
Yeah, no, that makes sense. I mean, it would be way too large to study. And I think you explained that brilliantly. So the trick here is we have to be able to identify the people that are chronically ill to define you have chronic Lyme by being able to evaluate that definitively, but also apply that same metric to people who aren't sick to say, hey, if you got Lyme disease, you would respond in a catastrophic way, and therefore you're high risk. And if you can identify the people that are on the predictive side, right, that would get sick if they got Lyme disease, and the people that are on the other side of it, that are the chronic side of it, we can have a better sample for testing to get better treatment for the chronic Lyme community is really what you're saying.
I want to break that into two things, because I was talking about testing people after they've had Lyme, and seeing how they're responding. But you're bringing up the point of, could we test you even before that? And could we see if you have an underlying vulnerability?
Yes.
I think that's also a very important question, because I know people who are very, very sick, who, if they had known that if they got COVID, they'd get long COVID, or if they got Lyme, they'd get chronic Lyme, they would have done things differently. And they would have protected themselves and taken different measures if they had known that they were at high risk. So I do think that knowing that underlying vulnerability is a very important question. I don't know if it's there. So that is one question that we're asking. We are asking this question of, are there people who, if they got, their immune system is poised to respond in a way such that if they got COVID in this context, or if they got Lyme in this context, they would respond badly? Maybe. Or maybe it's something about that strain of COVID, or that amount of COVID, or that strain of Lyme, or that amount of Lyme that triggered that response. And so maybe we'd only know after the infection has happened. These are questions. I'm always trying to ask questions in a way that makes as few assumptions as possible. But I also want to tell you about some of these bad responses. So when we looked at the Lyme biobank and the people who had, these are people who are over a year not recovered. Some of them are over 10 years not recovered. And one thing that we saw is a third of them were making really high levels of an antibody that we normally see in the context of allergy. So what does that mean? So this IgE antibody is normally identifying either a parasitic response or an allergic response. But we saw a third of people with chronic Lyme are making a lot of IgE that is specific to Borrelia burgdorferi. So what does that mean? Well, that's not autoimmune, because that's against a foreign bug. But it's also not a classic allergy, which is against something we call innocuous, right? Like allergies are normally against grass or peanuts, something that can't really hurt you. Your attack to it can hurt you, but the thing itself can't really hurt you, except that we have this mixed space of, you could be allergic to mold. Mold could hurt you, so could you be allergic to bacteria? And what happens then if you are now allergic to other spirochetes that live in your mouth? So these are self flora. They live with you. They live in your body. They're part of your bag of bugs, right? If you're now allergic to them, what does that mean? Is that autoimmune? It's not. You didn't have a break in tolerance. You're not attacking yourself, but you're attacking your bag of bugs. And what happens if you attack your bag of bugs? And I think.
And then it's constant, right, Dr. Tal? So essentially, you're exposed to grass and you have, an allergic reaction. You're exposed to a dog. You're exposed to a food allergen. But then the exposure dissipates and your immune system regulates. But in the case you're describing, the Borrelia is persistent in your oral, in your mouth or in your body or in your blood. So therefore, the immune response is persistent, causing chronic illness, right? Is what you're saying.
That's one context. And then think about this other context where maybe you had the Borrelia, you had doxycycline. Doxycycline actually cleared the Borrelia. The Borrelia is gone, but you still have Treponema denticola in your mouth, this other spirochete. And that IgE against Borrelia, well, the way that we looked at it, we saw that it binds to all spirochetes. So, well, now Treponema denticola lives in your mouth, and now you're attacking that all the time. So now what's going to happen? What does that look like if this thing you're allergic to never leaves you, and maybe you only feel better when you're taking antibiotics because you're killing these bacteria that you've become allergic to? I mean, this has led us to ask some really wild questions, like what happens if you become allergic to a bacteria?
Yeah, and it's wild because I didn't know that we knew about Borrelia. We've heard about syphilis, of course, because they're related, right? We've never heard about other spirochetal bacteria living in your mouth post Lyme infection.
You have multiple spirochetes that live in your mouth that are part of your plaque-forming bacteria. They usually don't like oxygen, and so they like to hide in your gum pockets. And you have several that are living in your mouth. The most famous of them is called Treponema denticola. You haven't heard of it, so it's not that famous, but you know, in the spirochetal world, it's one of the big ones, and a lot of people have that in their mouth.
That's wild. So I do want to ask a follow-up question about all this research you're doing, because one of the tools that you're using, or several of them, are around brain health, right? You're using brain check for cognitive assessment. You're using, I think, it's the WAVI egg or EEG, and that's the brainwave activity and reaction time. What are you seeing in the brains of chronic Lyme patients? We had a ton of Amen Clinic doctors on who told us some like wild stuff about how the brain changes structurally and from a activity standpoint because of Lyme. What are you seeing in your clinical trials using all the technology when assessing brain health of your patients?
Yeah. So in our study, one of the things that's been extremely profound has been reaction time. So no matter how we measure reaction time, whether we measure it through the eye tracking, how your eyes are moving, whether we measure it from that EEG cap, the WAVI, or whether we measure it on this video game, brain check, reaction time is slow in people with chronic Lyme and people with long COVID. And, you know, we are working with an amazing neuroscientist at Columbia, Marco Divina, who is, you know, really studying this with us and helping us understand, you know, reaction time is one of the ways that your brain, you know, this processing speed, right, that we're measuring in reaction time, this is one of the ways that your body is recognizing and manifesting that something is wrong, right? And so this is something that we are studying that is, you know, MAESTRO hasn't even completed yet. We're trying to get MAESTRO completed by the end of the year. What we are slow on is acute Lyme cases. If you hear of anybody who gets a tick bite, please send them our way.
Oh, wait, really? Acute?
Yes,
where do you have to live? Because I can send you a bazillion from Long Island.
Please. Oh my, if they're willing to come to MIT, they have to be able to come to MIT.
Okay.
But if they can come to MIT,
How long do they have to be there for? Just like one visit, or do they come back often?
One
Just
day.
day. That's all you need.
And then we are asking them to come back at 6 months and at a year.
Okay.
So it's 3 visits. And we ask a lot. As I said, this is surveys, physical assessments, biological samplings. We're asking a lot, but this is so key for us when we know the time of infection. And then we can ask these tests that we're looking at illness trajectories. We are trying to assess the accuracy of our ability to predict which way you're headed. And we have to have people at the onset of this infection in order to look at these trajectories, because everybody who's looking at what we're seeing in chronic Lyme and long COVID, they're saying, how is this all from that? You know, like with chronic Lyme, they're like, how can all of this illness just be from Lyme? You know, it has to be from other things, and it gets complicated, right? Like originally, when we were setting up the study, we were excluding people who'd had a concussion because we knew their brain was going to show signatures. We knew they'd have slow reaction time. We knew they'd have eye tracking issues. This is known for post-concussion, right? And so we were saying, okay, we're going to exclude people who've had concussions so we can focus on what happens after Lyme. But one of the things that we saw is that we were excluding everybody who had issues with something called proprioception. So your ability to identify where you are in space so that you can walk through a door without banging into it, and so that you can walk without falling on the floor and tripping down the stairs. This is this is proprioception. And what we saw is that there were all these people who wanted to enroll, but after they got Lyme, they'd fallen and gotten a concussion.
Yes.
Now they have these other things. And so we found ourselves saying, you know what? We're not going to exclude concussions because we don't want to miss everybody who's developing issues with proprioception. Yeah. However, we then still have to tease apart what of this is from the concussion and what of this is from Lyme. But the thing is, if Lyme caused a problem with proprioception, which then caused the fall, which caused the concussion, in some ways, it all traces back to Lyme. And so being able to do all of these measurements on somebody who's having that acute illness. They've just gotten the tick bite. They've maybe just had their doxycycline. They're in that first month of this, and we don't yet know which way they're headed. And we can measure everything as they were. And then we can track them over that next year and see where they end up at the end of the year. That's what really allows us to say, if we see that all these people who got Lyme are then having all these falls and concussions, and then we can say, you know what? These things that come from the concussion, that is part of Lyme, because the falls were part of Lyme. Because before they had Lyme, they weren't falling like this. So that's why this helps us so much.
Yeah, I can tell you it's just so interesting that a lot of your observations are also early observations we had to take boot camp, where we kept saying, why do so many people on this podcast tell us they've had one, and in most cases, many concussions? And then we realized, well, neurological Lyme disease, you're off balance. I've experienced it, right? And it's like, that's the reason they're becoming off balance. They can't really walk straight, and they're more prone to have a fall and get a concussion, which is super, super common in our community. So that can get kind of confusing and complicated. So I do want to say that we will certainly help you get people that have acute Lyme. Let's define that, right? So somebody gets bit by a tick, they know they were bit by a tick, they get a positive Lyme test in the same season, and now you want them to come to you and you want to basically study them. Is that a simple way of describing what you're looking for?
Within that month, ideally, yes.
Okay, in the same month of the diagnosis.
Exactly. We don't want to delay anybody starting treatment. We want you to start treatment right away. So we are not asking you to wait on that, but we would like you to come as soon as you can and participate in MAESTRO. Yep.
Will being treated prevent them from participating? Meaning if they get a diagnosis and they start treatment, they can still come while being treated?
They can still come while they're on treatment. Absolutely. We don't want anybody to delay treatment to participate in our study.
What if Rich and I find somebody and we bring them out there and we record the process and use that as part of our awareness tools? How would you feel about that, Dr.
That would be amazing, and we would love it.
We'll get to see you in person again.
If they're willing to be recorded, yes, 100%.
We will get you acute.
Yes.
Okay.
I can't tell you how many we have all the time. I mean, if we were in the spring or the early summer right now, we could bring you a whole bunch this week.
The adult ticks are biting right now. This is October is their season. So yeah.
We don't see them as aggressive in the fall as we do in the spring.
You don't.
Yeah. Yeah. Long Island. We don't, but yeah.
It's usually a smaller curve. Yep.
Yeah. So we have kept you for 2 hours.
Yes.
So I can't tell you how many other podcasts we'd love to do with you. I would hope at some point, and I'll leave this as a teaser for our community. At some point, I'd really love to do a Lyme and Lyme and female health podcast, because we have so many different questions that, again, I didn't know where we were going to go. And, we don't prepare questions for our guests. We go where the spirit takes us. But we've had so many guests on this podcast with female health issues that you've begun to answer for us. But unfortunately, I'm frustrated because there are so many more that I want to. So I'd really like to have a podcast just focusing on that issue. Another separate podcast I'd really love to do would be a podcast on the brain and the immune system. One of your
and you guys are gonna have Yuri Kim on.
Yes.
She is the conductor of MAESTRO. So she is our lead clinical study nurse, who's amazing. She comes from a background of research in Alzheimer's and years in the ER. She knows neurological illness, and she is the first author on our first publication that's going to be coming out of MAESTRO. We are in the process of finishing it right now on that reaction time story. And so she is going to tell you guys all about that. Okay.
So I'm looking forward to that. So one of your former colleagues, Andrew Huberman, had a podcast recently, an Essentials podcast on the brain and the immune system. And he raised a number of different issues. A lot of them would come, running through my brain while you were talking today that I'd really like to revisit with you, especially as it relates to Lyme. So as a teaser for future potential podcasts, and Matt and I would have you on every day because this is just fascinating stuff. And you've answered so many questions that we have had for years and years now. And I can't pay you a higher compliment than to say that despite doing, 5 years of interviews and interviewing more than 1,000 experts on Lyme disease, that you've answered so many questions that no one else has ever answered for us.
Yep.
So
Yeah.
thank you for, the great work that you're doing, and thank you for the really understandable way that you describe these really complex topics. But please consider coming back and doing at
And
least.
please consider coming to visit. I would love to have you guys record a local podcast
We
We're
at the
there.
lab where we run the study. I would love to have you guys see
We
all
will.
the fun tools.
So we will
that we
absolutely do that. So we do have we set up our studio actually at the ILAS conference, and we did 64 interviews in 3 days. So we actually have the ability to be mobile. So
So come on.
if
Yeah.
the invitation is there,
We're coming.
yes, we are there. We will take the ferry over and we will drive up and we'll
Perfect.
set up
And bring our cute Lyme patient at the same time. We'll kill two birds at one stone. We don't know who it is yet. That poor soul.
Well, so let's please let's spend some time planning our next podcast, which would be the Lyme and women. I don't know what the title will be, but I'd really love to do that piece. Because it really is lacking. There's just not enough, I think, conversation about that topic, and I just think we need to do that. So again, thank you. This is an awesome podcast.
Thank
We
you
really
guys
enjoyed
for
this, and I know
everything that you do and for all the awareness that you're raising. This is really critical. And there's so much that we do in lab that never reaches the people who need to hear it. So thank you